what is transpiration and how the transpiration is going to help the plant in their important metabolic activity. We will get all these answers in the presentation. This is Dr. Pankaj Kumar, your mentor for the current sessions. So in today's sessions, we will be talking about stomatal movement, right? So the first thing, we need to understand what exactly is the transpiration. Transpiration is the loss of water from the aerial part of the plants in the form of water vapor. So the three things is included. There should be a loss of water in the form of water vapor, right? And it should be from aerial part of the plants, okay? And it should be lost in the form of water vapor only. So these are the three things that is included with the transpiration. Now let's talk about the types of transpiration. 4 types has been recognized, what we call stomatal transpiration, cuticular transpiration, lenticular transpiration and bark transpiration, okay? So out of these 4, the maximally transpiration is going to occur from stomats only. And this is followed by cuticular, lenticular and the bark transpiration, right? So that is the sequence in which the plants participate in the role of function of transpiration. Now before we start discussing about transpiration, first of all we need to understand the structure of stomats. So in case of Dicot and Monocot, there are different stomats. For instance, if you talk about the Dicot, in case of Dicot, the stomats are actually bean shaped or kidney shaped, as you can see in the diagram as well. You see, this is a kidney shaped, and these are very, very specific with reference to the thickening. For instance, there is an inner thicker thickening and outer there is going to have a thinner thickening, okay? So due to differential thickening, actually it helps in opening and closing of stomats, right? And outside, these are the what we call, these are complementary cells, okay? Now if you talk about the monocot, in case of monocot, what happens that here the guard cell is a dumbbell shaped cells, you can see there, right? So in case of dumbbell shaped cell, what happens, this is a thinner part and this is the thicker part, right? So again, due to differential thickening, it helps in opening and closing of the stomats. But for a change, they don't have any complementary cells. So in case of Dicot, we have seen the complementary cells, but in case of monocot, they don't have any complementary cells, right? So let's try to understand that how the stomats are going to open or close. The very opening and closing of stomats is a function of the turgor changes in the guard cells. So there are 2 conditions. 1, if the guard cells is going to be turgid. If the guard cells is going to be turgid, what will happen? The thin wall get extended, right? Then the thick walls become concave and stomats are going to open. So turgidity of guard cells is required to open the stomats. But on contrary, if the guard cells become flaccid, what will happen? That it will lose water, the thick walls revert back to original position, right? And stomats are going to be closed. So we can see. So these are the turgor changes that is going to happen in the guard cells which affect the opening or closing of the stomata. So naturally, the metabolism is entirely depends upon the sequence of events that eventually do the changes in the osmotic concentration or the turgor properties of the guard cells. So we will try to understand it, okay? You see, there are different theories has been given, okay, and all those theories can be broadly divided into 2 groups, what we call classical theory and the modern theory. And even in the classical theory, the one of the thing that is the most famous, what we call sugar starch hypothesis, okay, and the most accepted theory is the theory of potassium ion transport theory, and that was given by Levitt. We will discuss it later. But first of all, let's try to understand that what exactly is a sugar starch theory and why it has been rejected, okay? So there are different scientists that has from time to time they have proposed their own hypothesis. So we will try to understand that what are the sequence of events that is going to happen during the sugar starch hypothesis. So this theory says that the photosynthesis is going to occur in light. As a result, there is a consumption of CO2. So once the CO2 is consumed, then the carbonic acid will not form. So that means the lowers H ion concentration of cell sap, or pH is going to increase. So at the higher pH, what will happen? That enzymatic conversion of starch into sugar will happen. How? Sugar in the presence of enzyme phosphorylase gets converted into glucose 1-phosphate. Glucose 1-phosphate in the presence of enzyme phosphoglucomutase gets converted into glucose 6-phosphate. And glucose 6-phosphate in the presence of phosphatase gets converted into glucose plus phosphate. So what we have seen, that a starch getting converted into glucose. So glucose will eventually increase the osmotic potential of guard cells. So there is going to have an endoosmosis, and as a result the guard cell will become turgid. Okay, and if the guard cell become turgid, stomats are going to open. So this is the simple thing as it is explained in case of the sugar starch hypothesis. Okay, suppose if you talk about the closure, so in case of closure, what happens? That in the dark there will be no photosynthesis, so that means there is accumulation of CO2, and if there is accumulation of CO2, then carbonic acid is going to be formed, then the carbonic acid will decrease the pH. So at this condition, what happens? That glucose will bind with ATP in the presence of hexokinase, they will form glucose 1-phosphate, right? Glucose 1-phosphate gets converted into starch. Okay, so what we see, that here at low pH the sugar getting converted into starch. Okay, so the exosmosis is going to happen. What will happen? That guard cell is going to be flaccid, and if the guard cell is going to flaccid, the stomata will be closed. Okay, so this is how opening and closing is going to happen. If I show it through the flow chart, we can show it like that, that uptake of CO2, then there is an increase in the pH, pH is getting a basic, so starch getting converted into glucose, osmotic potential of guard cells increases, endoosmosis, GC become turgid, and the stomats open. Similarly, during the dark, what happens? No photosynthesis, release of CO2, right? Then pH decreases, then glucose getting converted into starch, then osmotic potential of guard cell decreases, endoosmosis, exosmosis, GC become flaccid, and stomats closed. So this is how the closure and opening of the stomats has been explained through this classical starch-sugar hypothesis. So it is a combination of both the enzymes as well as the very concentration of glucose and starch. But what went wrong with this theory? Why it has been rejected? You see, the very first drawback was that starch-sugar conversion is very slow. It is slow to such an extent that it cannot influence the requisite changes in the osmotic potential to do exosmosis or endoosmosis. One. Number two, some plants like monocots, the starch is not there. So where is the question of starch and sugar interconversion? So again, the theory fails to explain the stomatal movement in case of monocots. One more thing, glucose is not at all detectable, or even if it is detectable, it is in a very, very small quantity in case of the guard cells. So again, this theory doesn't hold true. So on all this count, this theory has been rejected, right? So this was all about the sugar starch hypothesis. In the next sessions, we will be talking about the actual transpirational theory, that is active potassium ion theory, in the next class.